Hello everyone, welcome to my YouTube channel Biochemistry Simplified. We are starting a lecture series on medical biotechnology. What is the meaning of medical biotechnology? Technology means application of science for betterment of human life, and when we say bio means the application of biology for betterment of human life. Medical means anything that is related to human health. So, medical biotechnology means application of biology for betterment of human health. And how to make the human health better? The human health is made better by better prevention, better diagnosis, and better treatment of diseases. So, in simple terms, medical biotechnology means the application of biology for prevention, diagnosis, and treatment of diseases. In medical biotechnology, we use a biological system, a living organism, or a part of living organism or biological system to develop useful products. For example, we use some bacterial cell, some human cell, or maybe some molecule like protein, DNA, and so on. So we use a biological system, a living organism, or a part of biological system to develop useful products. And we develop the useful products for what? We develop the useful products for prevention, diagnosis, and treatment of diseases. I'll give you some examples how medical biotechnology has helped us in better prevention, diagnosis, and treatment of diseases. With the advancement of medical biotechnology, we have developed vaccines, we have developed monoclonal antibodies for prevention of various diseases. For better diagnosis, also, we have developed RT-PCR, which is a household name these days, and we have developed DNA fingerprinting. For treatment, gene therapy, nanotechnology, and biotechnology even helps in the research like protein sequencing, cloning, even genome mapping. So these are some examples where biotechnology has helped us. There are two sciences which form the base of biotechnology. One is molecular biology and the second one is immunology. These are the two basic sciences which form the base of biotechnology. And there are two techniques in the core of biotechnology, that is chromatography and electrophoresis. So biotechnology is based on two sciences, molecular biology and immunology, and all the techniques in biotechnology are based on chromatography and electrophoresis. So we will discuss basics of chromatography and basics of electrophoresis, and then we will discuss the individual techniques. Let us start with chromatography. Chromatography started in 1901. In 1901, there was a Russian scientist called Mikhail Svet. So Mikhail Svet, he took a glass column, filled it with calcium carbonate, and then he took plant extract dissolved in petroleum ether and ethanol solution. And after that, he poured this solution, this plant extract, onto the column filled with calcium carbonate. Now, this plant extract, as it is percolating through this column, over a time period, the colored bands were produced. And when these colored bands were analyzed, it was found that the uppermost band, which was brownish in color, was due to the presence of carotenoids. The second band, which was yellowish in color, was due to the presence of xanthophyll. The third band, which was greenish, that is light green in color, was due to the presence of chlorophyll A. And the fourth band, that was dark green in color, was due to the presence of chlorophyll B. So all the plant pigments have been separated and they have produced colored bands. So this technique was given the name chromatography. Chroma means color and graphy means design. And Mikhail Svet is known as the father of chromatography. So this was the beginning of chromatography way back in 1901. In chromatography, basically we separate similar kind of substances. So it is a technique for separation of similar kind of substances from a complex mixture dissolved in a fluidic solvent. For example, a mixture of amino acids, a mixture of monosaccharides, a mixture of lipids, and so on. In chromatography, there are two phases, the stationary phase and the mobile phase. Stationary phase is the material fixed on a system, and the system can be a column, like in the Swett's experiment. It can be a plate, or it can be a sheet, means a glass slide, or a plastic sheet, or a aluminum foil, or a column where some material is fixed, that is acting as a stationary phase. Mobile phase is basically the fluidic solvent in which the molecules are dissolved. So this fluidic solvent is carrying the mixture along with its constituents through the system. As the mobile phase moves through the stationary phase, the different constituents of the mixture, they have different affinities for stationary phase and for the mobile phase. So those molecules which have more affinity with the mobile phase, they will move faster. They will be going along with the mobile phase, so they will be fast. But those molecules which have more affinity with the stationary phase, they will stay with the stationary phase and they will move slower. So over a time period, the molecules get separated due to their differential affinities for the stationary phase and for the mobile phase. This is the basic system of chromatography, the basic principle of chromatography. I'll give you some example. Like in the paper chromatography, we take a filter paper sheet, and suppose this is an amino acid solution. On the filter paper sheet, we make a baseline, and on this baseline we put a drop of the amino acid solution, and after that we suspend this paper in a jar containing a solution of butanol, acetic acid, and water. Now this solution rises upwards due to the capillary action, and when this solution is rising upwards, the paper, the chromatography paper, the filter paper, is basically cellulose, and cellulose is containing all the hydroxyl groups. The water gets associated with the hydroxyl groups to form the polar stationary phase. Butanol is a nonpolar molecule, so it keeps moving upwards, so that forms a nonpolar mobile phase. So there are two phases now, the polar stationary phase and the nonpolar mobile phase. As the solution is moving upwards, it is carrying the amino acids along with it. So those amino acids which are polar, they have a tendency to associate with the polar stationary phase, so they will move slower. And those amino acids which are non-polar in nature, they have a tendency to associate with the moving butanol, with the moving non-polar phase, so they will move faster. So over a time period, these amino acids are separated. And they are separated on which basis? They are separated on the basis of their polarity. The non-polar, the most non-polar being the fastest, and the most polar being the slowest. So this is a very good example of the chromatography, how the molecules get separated. Now these amino acids have got separated, but they are not visible. To make them visible, ninhydrin is sprayed on the filter paper sheet, and this ninhydrin forms a purple colored complex with the amino acid. So this is what we finally get. All the purple spots are belonging to some amino acid. But how do we know which which spot is belonging to which amino acid? One way is that we run a standard amino acid. Along with the mixture of amino acids here, we run a standard amino acid here. For example, we run leucine here, and suppose leucine has moved up to here, means this spot is belonging to leucine. That is one way. But practically speaking, this is not possible every time. So what we do, we have to calculate the RF value. What is RF value? RF value stands for the retention factor. Now this is the distance traveled by the solvent front, and this is the distance traveled by amino acid number 1, amino acid number 2, amino acid number 3, amino acid number 4, and so on. So RF value is the distance traveled by the amino acid and distance traveled by the solvent front. and every amino acid has got a different RF value. This is just an example of the RF value of different amino acids under standard conditions. So this is one very good example of chromatography, the paper chromatography. There is another type of chromatography which is similar to paper chromatography, that is called thin layer chromatography, or TLC in short. In TLC, we take a plastic, glass or a metal foil plate which is coated with the material, that is S for silica, A for alumina, C for cellulose. To remember it, you can remember it as sac. So sac is pasted, a thin layer of sac, that is silica, alumina or cellulose, that is spread over a plastic, glass or metal foil plate. And rest of the things are same as paper chromatography. This plate is suspended in a solution, and as the solution is rising upwards, the molecules get separated. Same as paper chromatography, but this is advantageous over paper chromatography. So TLC has advantages over paper chromatography, that is multiple samples can be run simultaneously. It is faster than paper chromatography. It is better, qualitatively better, or you can say the separation is better in TLC as compared to paper chromatography, and even the quantitative analysis is better in TLC as compared to paper chromatography. And the choice of adsorbents, in paper it is just a sheet of paper, but here we have different adsorbents, silica, alumina, cellulose, so that gives us a wide range of the materials which we can separate on TLC, and there is less chance of cross contamination. These are some advantages of TLC over paper chromatography. What the best use of TLC is for screening for drugs of abuse, like performance enhancing drugs in sports, or even for therapeutic drug monitoring. So we can simply remember drugs of use and abuse. So basically for the monitoring of the therapeutic drugs, or for screening of the contraband or the illegal drugs. So drugs of use and abuse. That is the point to remember. What is the use of chromatography? There are two uses of chromatography. We can say there are two types: preparative or analytical. Preparative chromatography means to purify a substance. Analytical means to detect the presence of a substance in complex mixture. And the analytical chromatography not only detects the presence of substance, it also measures the small quantity of substance in a complex mixture. So these are two uses of chromatography, that is to purify a substance or to detect the presence of a substance, or we can say to measure the small amount of substance in a complex mixture. And these two are not mutually exclusive. Paper chromatography is preparative as well as analytical at the same time. Important points to remember from this discussion: the father of chromatography was Michael Sweat, who separated the plant pigments in 1901. Paper chromatography separates amino acids on the basis of polarity, most nonpolar being the fastest. TLC, the use of TLC is for measuring the drugs of use and abuse, detecting and measuring. RF value is retardation factor, and to memorize it, we can memorize it as ratio of fronts. Which fronts? The distance traveled by the individual amino acid and the distance traveled by the solvent front. That is RF value. The different It is different from retention factor. There's another term called retention factor which is used in column chromatography. Retention factor means the time the substance is retained in the stationary phase upon the time the substance is retained in the mobile phase. Now it is an entirely different entity. Okay, so these are the points to remember. So with that we end our first lecture on biotechnology, and if you like the lecture, please subscribe and like it. Thank you.